Ladies and gentlemen, dear teachers and students, good afternoon. Welcome to the lectures of the Great Master Series of Science and Technology. I'm today's host, Xu Xingyi, from the MUST Art Troupe. Today, we are very honored to invite Dr. David Ho, Professor of Microbiology and Immunology, and Helen Wu, Professor of Medicine and Director of the Aaron Diamond AIDS Research Center, to bring us a lecture on addressing viral pandemic. First of all, please let me introduce today's guests. They are Dr. Liao Zeyun, Chancellor of Macau University of Science and Technology. Chair Professor Joseph Hong-Wei Li, Vice Chancellor and President of Macau University of Science and Technology. Dr. Chen Jiming, Secretary General of the University Council of Macau University of Science and Technology. Chair Professor Paul Tan Guangheng, Vice President of Macau University of Science and Technology, member of the Hong Kong Academy of Sciences. Mr. Lin Jiawei, member of the University Council of Macau University of Science and Technology. Chair Professor Zhu Yizhun, Associate Vice President and Director of School of Pharmacy of Macau University of Science and Technology, member of the European Academy of Science and Arts. Professor Liang Wenhui, Associate Vice President and Dean of Liberal Arts of Macau University of Science and Technology. Professor Chen Weijian, Associate Dean of Tsinghua Shenzhen International Graduate School and colleagues. Mr. Zhao Liang, Vice President of Guangdong Hengqin Jintou Venture Capital Fund Management Co. Limited. And many other guests from our university and other institutes in mainland China and in Macau. Welcome, everyone. First, we would like to invite you all to take a group photo together. So we will ask the photographer to the stage. Please look at the camera. Ready? 3, 2, 1. Thank you. May we have the honor to invite Chair Professor Tan Guangheng, Vice President of Macau University of Science and Technology, to deliver a welcome speech.
Dear distinguished guests, teachers and students, good afternoon, everyone. Today, we are honored to have the renowned physician-scientist, Dr. David Ho, Professor of Microbiology and Immunology, Founding Scientific Director of the Aaron Diamond AIDS Research Center, Clyde and Helen Wu, Professor of Medicine at Columbia University Irving Medical Center, as the keynote speaker for this semester's Great Master of Science and Technology lecture series in medicine. Dr. Ho is a member of the US National Academy of Medicine and American Academy of Arts and Sciences, a fellow of the American Association for the Advancement of Science, and an academician of the Chinese Academy of Engineering. Dr. Ho achieved global fame when he was named Time Magazine's 1996 Man of the Year. Time acknowledged in 1996 that Ho is not, to be sure, a household name, but some people make headlines while others make history. So how did Dr. Ho make history? After receiving his degrees from California Institute of Technology and Harvard Medical School, he has been at the forefront of AIDS research for some 40 years since then. His elegant studies unraveled the dynamic nature of HIV replication in vivo, and revolutionized our basic understanding of this horrific disease. His introduction of the combination antiviral therapy instead of the prevailing single therapy in the 90s was the turning point in the AIDS epidemic, transforming an automatic death sentence into a manageable disease. Since the start of the COVID-19 pandemic, Dr. Ho's research team has devoted considerable efforts to develop novel strategies to diagnose, treat, and prevent SARS-CoV-2 infection. There are many lessons learned to be shared from both AIDS and COVID-19, and such knowledge will undoubtedly be applicable in addressing future viral epidemics. The topic of Dr. Ho's speech today is therefore addressing viral pandemic. We're looking forward to the wonderful sharing from Dr. Ho. Let's welcome Dr. Ho. Thank you.
Thank you, Professor Tan. Please be seated. A simple but grand souvenir presentation ceremony will be held below. May we have the honor to invite Dr. Ho to the stage. May we now invite Dr. Liao Zeyun, Chancellor of Macau University of Science and Technology, to present the gift to Dr. Ho and take a group photo together. Please look at the camera. Thank you, Dr. Liu. Please be seated. And Dr. Ho, please stay. Now the lecture begins. Let's welcome Dr. Ho.
Thank you very much. Good afternoon, everyone. Let me begin by congratulating Macau University of Science and Technology for the occasion of your 24th anniversary. Congratulations. And let me thank the leadership of the university for the honor of an honorary doctorate in medicine. I am very grateful. I also like to thank all of you for a wonderful but large lunch. Now most of the blood is going to my digestive system rather than my brain, but I'll do my best. I'm going to tell you about my work addressing two different viral pandemics. And here are the two viruses that I have dealt with over a career spanning about 43 years. The first is HIV that causes AIDS. It's a virus that's infected about 70 million people worldwide, half of whom have died already. But the other half could be treated and managed and probably have a normal lifespan. So I'm going to spend the first half of my presentation not so much presenting a technical talk, but I'll tell you the story of my experience through that particular journey. The second one on SARS-CoV-2, which is the causative agent of COVID-19, I've worked on for the past 4 years because I think everyone recognized the devastation that we all suffered through that pandemic. It's still fresh in our minds. By our best estimate, at least 3/4 of the world's population were infected, and over 7 million people have died, at least documented, probably a bit more than that. So, and that will be a bit more of a scientific presentation. So let me begin with this in 1980, when I was finishing my medical training in the west part of Los Angeles, and this patient came to the hospital with pneumonia, as you see on the left, and with the skin lesion, as you see on the right. And it turned out to be a young man who had pneumocystis pneumonia and Kaposi sarcoma, two conditions that normally would not plague a person with an intact immune system. Yet this previously healthy young man was coming down with a very unusual disease. He was treated, sent home, but died a few weeks later, to everyone's surprise. And then in rapid succession, over a course of a few months, we saw another and yet another. And these initial 5 cases formed the report by the U.S. Centers for Disease Control, or CDC, that described what we now call AIDS. So that was at least the visible beginning of the AIDS pandemic reported in 1981. And as I was finishing my medical training and about to go enter a research career, already focusing on viruses, instead of working on other viruses, I thought this new disease might be caused by a viral agent, and therefore began my research at the Massachusetts General Hospital in Boston, working with 2 mentors, Marty Hirsch and Chip Schooley. And immediately I tried to find the agent that was responsible for this mysterious and yet new disease. Of course, I failed while working on it for a couple years. And the virus was initially identified by François Paré-Sinoussi as well as Luc Montagnier at the Pasteur Institute in Paris. They saw the initial evidence of a virus, in fact, a retrovirus. But it was not sure that this was the cause of AIDS. But a year later, Bob Gallo and his colleagues at the NIH made the definitive diagnosis of the causative agent. At that time, it was called HTLV3, and of course, now it's called HIV. So from that point on, it was known that this virus was responsible for this horrific new disease. But being a young, opportunistic scientist working in this area, I was able to quickly contribute to the field by first showing that healthy people actually could carry the virus for a long time, so-called asymptomatic state. And in fact, we could find the virus in blood and in genital secretions. From there on, still working as a research fellow, along with my colleagues at Mass General, we were able to demonstrate that in addition to attacking the immune system, the virus is also capable of attacking the nervous system, causing dementia, neuropathy, spinal cord degeneration. Those complications we seldom see today, yet in the early days of untreated AIDS, they were extremely prevalent and extremely devastating to the patients. Then a bit later, by the late 1980s, we started to find that contrary to earlier belief, the virus is actually quite commonly detected in blood. As the technology improved, we could detect more and more of the virus in the blood. Remember, after infection, there's no disease typically for many, many years. And that gave the impression that the virus wasn't doing very much. But as we find more and more virus, we find that perhaps the virus is actually, despite a relatively latent clinical state. The virus is not latent. And this was the first initial indication, and we went on to prove that, as you'll see. But by this point, in the late 1980s, I was asked by this philanthropist named Irene Diamond, who wanted to set up a new institute in New York City. And despite my young age, she recruited me to become the director of this new institute called the Aaron Diamond AIDS Research Center in New York City, which was officially formed in 1990. And I've been the founding director, and I'm still the director today. And this institute, for many, many years, was a part of the Rockefeller University until the year 2000. And I will tell you more about that. So our new institute began. I recruited a number of younger faculty members to join, and that's the beginning of our research institute. But by 1993, I had a chance encounter with this young man on the slide. He's no longer a young man, but Dale Kempf was a chemist, and he was working in a drug company called Abbott. And we had gone to the same meeting in the US, and we were both waiting in the car rental line and realized we were going to the same place, same meeting. And so we decided to share the car rental and talk all the way for about 2 hours. And it turns out he was working on drugs that would target a particular enzyme of HIV called the protease. And he's a chemist without the virology support. So starting in 1993 or so, we began a series of collaboration that led to some of our most important scientific findings. And make a long story short, we very quickly discover a new drug against the protease enzyme of HIV. At that time, it's ritonavir. It's not used very much nowadays, but it blocked protease, and we were able to show that it actually controlled the virus more potently than any prior agent tested. So that was a great success in terms of new drug development. However, that drug actually became an extremely useful tool to understand HIV dynamics, which Professor Tam just talked about. So what we have noticed by that point in time was that in an infected individual, there's almost, as you look at the virus level in the blood, there's a very steady level for not only days and weeks, but for months and years and sometimes decades. That's what we call steady state. And the steady state actually is very important because if that level were to be high, the prognosis is very bad. If that level were to be low, the prognosis is much better. However, what's going on during that steady state? Well, I don't need to tell the students. If you have a steady state, that means the amount of virus produced that time is equal to the virus that's cleared. That's what steady state means. However, if you have a sharp instrument like the new drug that, when given, abruptly blocks HIV replication and you take away production, if you just follow the decay curve of the virus, you could actually comprehend the dynamics. Now, I think most of the students here have taken high school calculus and perhaps freshman calculus. So you could write two differential equations, one for the cells that are producing virus and one for the virus particle themselves. and you could fit the data to those equations and derive kinetic information. And that's what we did by mid-1994 and presented the results in January 1995. And what was found is that the decay, once the drug is administered, is extremely rapid. And that means during the steady state, the production is equally rapid. And that tells you that how dynamic HIV was. And in fact, from that initial study, we could tell that billions to trillions of virus particles were made in the body of an infected person every single day. That would go on for years and even decades. And associated with that is a rapid destruction of a critical cell in our immune system called CD4 T cells. We published that study, and then it turns out that concurrently, another group led by my friend George Shaw at another university made exactly the same observation, and we published the two papers together. My team then went on. In fact, we worked with Alan Perelson, who's a mathematician at Los Alamos National Laboratory, to define the dynamics even more precisely. I won't go into the specifics because this is a very general, diverse audience, but we've worked out the dynamics of HIV replication to great detail in these two publications. But the most important thing, obviously, is the implication of the dynamics for treatment. And I will, and that was already clear to us after our initial study on the dynamics. And I wrote this particular commentary to suggest that we need to change the paradigm for AIDS therapy. We need to treat it early, and we need to treat it aggressively. And the rationale, I think, will be clear in a few videos that I'm now going to show you. And these were taken from AIDS documentaries.
I don't think that people can understand what it was like to live in New York in the 80s and early 90s. The gay community here in New York was completely devastated by HIV infection, and being a member of that community, I was basically a doctor by day, a nurse by night, and a mourner on weekends. I mean, it was just unbelievable.
Markowitz took a job with AIDS researcher David Ho, who was trying to unravel one of HIV's greatest mysteries. Why did the virus seem to lie dormant for years?
Throughout the 1980s, we knew quite well that HIV could infect the person and not cause disease for many years, in fact, as long as a decade or more. And that lulled us into thinking that HIV wasn't so active.
That's me.
Dr. Ho and other researchers began using new technology to measure the amount of virus in a patient's blood. They discovered that the virus was never dormant. From the very moment of infection, HIV was at war with the body's immune system.
What was realized is that HIV is a very dynamic infection, not an infection that's latent.
Scientists discovered that HIV replicated by the billions every day, and as it did, it mutated.
HIV is a virus that doesn't make copies of itself very well. It's sort of like a little child typing at a typewriter, makes lots of mistakes. And the combination of lots of mistakes and a high turnover rate made for a lot of mutations. If you have a lot of mutations, then drug resistance is a big problem.
So far, the virus had developed resistance to every known drug, like AZT. Ho and his team were among those working on a radical new treatment.
We came to the conclusion that it's inevitable for HIV to develop drug resistance if you give it one drug at a time. However, if you start to combine the drugs and try to force the virus into a corner using multiple drugs, it is exceedingly difficult for HIV to become resistant to all the drugs simultaneously.
3 drugs, including the most advanced called protease inhibitors, would attack the virus, boxing it in.
Excuse me. Excuse me, it's a little warm up here. So, and with the combination therapy, these are the results we saw. In red, you could see the viral level drop so dramatically and is durably controlled for many months, beyond a year or 2 even. And the immune cells shown in the blue, CD4 T cells, actually increase, although do not typically return to normal. And this is the turning point of the AIDS therapy. So let me play another. I will.
In 1996, at a global AIDS conference in Vancouver, researchers announced the results of their clinical trials to the world's media.
As we expected, with this triple regimen, we put out the fire, in the sense that when we look in the blood for evidence of viral replication, we cannot find it.
I think the experiments I talked about, the ones Marty Markowitz talked about, are trying to determine the feasibility of eradication.
It was called the triple cocktail. Time named David Ho Man of the Year. With these new antiretroviral drugs, HIV infection was no longer a death sentence.
The people who were on disability got out of bed and started looking for things to do, and the funerals stopped and the inpatient units emptied out. It was a true miracle.
By the way, that was my colleague of many, many years, Marty Markowitz. So at the end of this international conference meeting, a very dramatic moment occurred. A woman, as you'll see, from Africa stood up and basically said, "You're celebrating all this success, but what about us?" And let me play that video as well.
At Vancouver, the news had been about the scientific breakthrough, not what the drugs would eventually cost, as much as $16,000 per year.
Catherine Mary and lives
At the close, a Zambian mother addressed the delegates. She was dying of AIDS.
Thank you very much for providing my accommodation while in Vancouver, which cost the equivalent of 3 years' rent for me.
She posed it quite simply, that what does this mean for us? Our fate is the same.
Thank you for my air ticket, the cost which would feed my children from now until they reach adulthood. God willing, thank you very much for listening.
And this, at least to me, shook up the field. We need to do not only research, but make sure whatever the fruits of our research could be delivered to those in need. And her remarks, I think, was the very beginning of the effort that then led to many foundations, such as Gates and Wellcome Trust Foundations, and the Global Fund set up by the World Health Organization, and later the U.S. program called PEPFAR, or President's Emergency Program for AIDS Relief. And it is through these programs now that effective combination antiretroviral therapy could be delivered to the world. And gratifyingly, now approximately 2/3 of the patients who are infected and carrying HIV could be on therapy. And most of that 25 million people on therapy are in sub-Saharan Africa. So as the years went on, you could see that was 1996. We spent years follow-up trying to develop HIV vaccines. Two of my colleagues, Huang Yaoxing and Chen Ziwei, actually made two different vaccine constructs over many years. We took both to what's called GMP manufacturing, so we could test the vaccine products in patients. And we did that for almost a decade. But similar approaches completely failed. For example, the one developed by Merck, the US pharmaceutical company. And that shook up the field, and most of us who had vaccine in development did not have the confidence to carry forward. And so it was a most frustrating experiences that we all went through. At about the same time, it was being shown that, in fact, the same antiretroviral drugs could be very effectively used to prevent HIV infection. So, and this is done by a consortium of scientists, that if you give antiretroviral drugs every day to people who are not infected but are at high risk, in fact, you could prevent HIV infection with efficacy greater than 90%. And so that has been termed pre-exposure prophylaxis, or PrEP, which now is widely applied and has helped to reduce HIV transmission rate globally. So our research turned in this particular direction, but it is difficult to take a pill every single day when you're not infected. And so my colleagues and I switch our attention to focus on longer-acting approaches to HIV prevention. So one is shown here. In fact, we had developed a monoclonal antibody that could block HIV infection. And in this particular paper, we engineered that antibody to be even more potent and broader. And this antibody now is in phase 2 clinical studies. We also worked with scientists at GlaxoSmithKline, another pharmaceutical company, to develop, instead of daily pills, injectables that would last for a long time. And when injected, could prevent infection of related viruses in monkeys. And so this now is a drug called cabotegravir, which is given every 2 months and is very effective in preventing HIV infection in high-risk individuals. And then lastly, my colleague, Huang Yaoxing, designed and engineered bispecific antibodies. So one arm attacks one target and the other arm another, and came up with an antibody that is the most potent and the broadest against HIV. And this product is now gone through phase 1 and is in the process of being tested in phase 2 studies in Tanzania. So these are some of the things that we have been doing in HIV for nearly 40 years. Then by the year, it's not moved, okay. Our whole institute was moved from Rockefeller to Columbia. And we moved in January 2020 with brand new facilities and a brand new biosafety containment laboratory. And this is when, as you know, COVID struck, certainly in late 2019 in Wuhan, and by early 2020, much of the world. Our first case in New York City occurred on March 1, 2020. But by then, our research team already made the commitment to try to make a contribution to this new pandemic in late January 2020. So I'm just going to tell you, we went on to do many things, initially related to coming up with monoclonal antibodies. So we published 3 papers. Then, using these antibodies as well as sera from infected individuals, we went on to characterize how the virus evolved over time. From the original strain in Wuhan to alpha, beta, gamma, delta, and omicron, and many omicron subvariants. So we published numerous papers on the subject. At the same time, we utilize our expertise on the viral protease to look at how this new virus, SARS-CoV-2, evade protease inhibitors, a drug called nirmatrelvir. We published 2 papers on the subject, and then a paper on a broadly protective antiviral agent. I won't have time to talk about the protease inhibitors. In fact, I won't have time to cover much detail, but I wish to convey to you an impression about how SARS-CoV-2 has evolved, and a little bit about this, if there is time at the end, about the protective agent. So this is, on the left is an electron micrograph of SARS-CoV-2 with a spike protein extruding from the viral surface. And that spike is shown on the right. And that spike is actually a homotrimer with the receptor binding domain shown in the red and the N-terminal domain shown in the orange color. And of course, the receptor binding domain, also known as RBD, needs to pop up in order to engage the cellular receptor, the human ACE2 protein. Now, let me tell you about the monoclonal antibody effort. They were isolated largely by Liu Lihong, and the structural and the bioinformatics done by Guo Yichen, and the electron microscopy, cryo-electron microscopy was done by Ryan Kastner and Larry Shapiro. So to date, we have isolated about 1,500 monoclonal antibodies directed to the spike protein. And of course, in the very first study, we discovered about 20 that were extremely broad and potent. And they largely bind to the RBD and the NTD, with the antibodies directed to the RBD being much more potent than those to the NTD. And so this is our initial discovery, but of course, as you'll hear, these antibodies were then knocked out by the emergence of subsequent SARS-CoV-2 variants. But we found out from all the initial work that most of the antibody in the serum of infected individuals or vaccinated individuals are directed to the spike, and in particular, those directed to the RBD are responsible for much of the virus neutralizing activity. Only a minor portion is accounted for by antibodies directed to the N-terminal domain, or NTD. And most of these antibodies inactivate infection via blockade of the receptor. And in particular, those directed to the RBD could be grouped into 4 classes, depending on which surface of the RBD is being bound by the antibody. And I will simply refer to them as class 1, 2, 3, 4. So we went on to discover another antibody that is of great interest. And this is the antibody which we call 1040. It's a class 4 RBD antibody, and you could see the footprint of the antibody on the bottom right. And what's interesting about this antibody is that it actually not only neutralized SARS-CoV-2, but it could neutralize the original SARS virus from 20 years ago and many of the related viruses in bats. And so this antibody is very important in the future should another virus emerge from the bat from the same family. Of course, this particular one was knocked out by the emergence of Omicron variant of SARS-CoV-2. So we went on to characterize another monoclonal antibody, which I like to point out. And this one is actually not binding at the top of the spike, but it's binding, as you could see on the left here, with the orange portion, it's binding to the side of the RBD. And one wonders how it could neutralize HIV when it's binding to the side, because not HIV, SARS-CoV-2. And so it's not interfering with receptor binding. And so we went on to characterize the mechanism by cryo-electron microscopy and molecular dynamic studies, as you see here. So you could see on the top is a top view of the spike with 3 antibodies bound. And the green portion is breathing, but it's not moving. And here on the bottom, you have 2 antibodies bound. So there's one RBD that's moving up. And it appears that this antibody is inhibiting the up motion of the RBD and therefore locks the RBD in the down position, and the virus can no longer infect a cell via the receptor binding. So mechanistically, it's very interesting. So we use these antibodies, and as I said earlier, we use sera to understand SARS-CoV-2 evolution. And on the top is the global pattern, and then what is seen in Asia, Europe, and North America. And as you could see, in the first year, much of the virus that circulated, was detected, is the original virus from Wuhan. Then you see in late 2020, a variant evolved from southern England now called the Alpha variant. And the Alpha variant replaced the original variant in the course of a few months. And then during that period, the Beta and Gamma variants emerged from South Africa and Brazil, but those 2 never gained traction. But you see in green, by much of 2021 on, the Delta variant dominated. And that came from India and basically very rapidly took over until late 2021, when a variant emerged from southern Africa, which now we call Omicron. Omicron BA.1 is the first version of Omicron. And that extremely rapidly replaced Delta. And as you could see, BA.1 is quickly replaced by BA.2, then BA.5, then BQ, XBB. It becomes extremely complicated, right? And nowadays, about 95 to 99% of the virus found in infected individuals throughout the world is accounted for by the newest variant called JN1. So the virus is evolving almost in real time as we see it, and we've been, as a research team, trying to characterize this viral evolution a little bit behind, but almost in real time as well, and I'll tell you that experience. And here you see more closely what has happened over the last few months with the JN1 subvariant of Omicron basically taking over. Now, this is a phylogenetic tree, or a family tree of SARS-CoV-2. And if they're further apart, it means they're more genetically distant. And you could see the original variant shown in the upper left, wild type. And then it quickly emerged into Alpha, Delta, Omicron, Omicron BA2, BA5, BQ, XBB, EG5.1, and then more recently BA286 and JM1. Basically, the virus has evolved so much with so many mutations throughout its genome, and most of them occur on the spike protein. Now, the first vaccine that was made, of course, was based on the original Wuhan strain. But by the time that vaccine came out and widely administered, the Delta variant was already dominant. And then the next vaccine, at least in the US, is based on BA5. But by the time that vaccine came out, XBB and EG5.1 viruses were dominant. And so the vaccine is always behind the virus by about 6 months. And more recently, the vaccine is based on XBB 1.5 variant, as shown on the third arrow. But as you see, what we now have is JM1, which is genetically very, very distant from the current vaccine. So we're constantly chasing after viral evolution, always a step or two behind. And the evolution, you could see the mutations accumulate on the spike, particularly in the RBD. And you could see now that for JM1, there are about 50 amino acid substitutions or changes in the spike compared to the original strain. So this is very dramatic viral evolution. And we've been characterizing this. The first alpha and beta variant was characterized by a postdoc named Wang Pengfei, who's now an assistant professor at Fudan. And then the next variant that we characterized was actually something called the New York variant, together with our colleagues from infectious diseases. And then we characterized Pengfei also characterized the Gamma variant for Brazil. But the most dramatic variant that occurred is actually the Omicron. I think the world knows that. And this is amazing. In the US, it was in late November that WHO announced this variant with many, many mutations in the spike protein, particularly concentrated in the RBD domain. And these two colleagues, Li Hong and Shou, immediately jumped into action because we knew just by looking at the mutations, it was going to be a problem. And so the WHO announced this variant on November 24th. It took us about 10 days to have the gene synthesized, okay, because of so many mutations. But it only took 4 days for us to do the experiment. and for another week before we finished the paper. And the manuscript was submitted and actually was published on all within 1 month. This is a record that I'm pretty sure our lab will not break again. But the Omicron variant was dramatically different. And then what happened was, this is the subsequent BA2 variant. We characterized then, Wang Chen took over and characterized BA5, BA275, BQ and XBB, and more recently BA286, which is the immediate precursor of JN1. And I can't cover all the data in these papers, but I wish to convey to you the overall impression of virus evolution for this agent. So this is a summary plot of a lot of data. On the y-axis, we're looking at fold change in serum antibody neutralization. And sera were obtained from infected person or vaccinated person or a person with both infection and vaccination. On the x-axis is simply the change in receptor binding affinity as measured by SPR. And you could see in general the viral evolution move into the right upper quadrant, right? And this may be surprising. You would think the viral evolution would occur to evade antibodies, so you would expect full increase in antibody resistance. But you would think the virus would lose some affinity for the receptor, but instead it gains receptor affinity. But most dramatically are, if you look, the dominant viruses that are the latest are the ones that are most antibody evasive. So largely, the evolution is driven by antibody pressure. So this is another way to plot virus evolution. So this is actually what we call antigenic cartography. It is every square, every unit is twofold change in antibody resistance. Okay, and you see the original wild-type virus there in black. And we could show that those that are clustered by the gray areas represent linear mutation, stepwise accumulation of amino acid substitutions. And so that's not surprising. You expect that to happen. But what through our analyses, what is most interesting is the huge leap from the wild type to BA1. So that's the initial Omicron. That's a huge antigenic jump that just occurred like that. And interestingly, that came out of Southern Africa. And the same with BA2, jumping to BA286, that became JN1 on the right bottom. That's an even larger jump. And that too came from Southern Africa. And why? It suggests that through one individual, or maybe a few individuals, the virus was able to replicate for a long time and accumulate mutations for a long time. And Southern Africa has the highest prevalence for HIV infection. And virus is able to replicate for that long in immunocompromised individuals. So events like this, stochastic events, saltatory events, pretty much unpredictable. So, there are a lot of people using AI trying to predict the future trajectory of SARS-CoV-2 evolution. But to me, these are black swan events that are difficult to predict. In addition, two different viruses infecting a same person could recombine and create new ones. And that's exemplified by XBC viruses, and importantly, XBB, which was very dominant for a period, was the result of a recombination of two different viruses that occurred somewhere in the United States. So that too is very much unpredictable. So people often ask me, where's this virus going? And my answer is, I don't know, because of events like this. So let me give you another example of how the virus has evolved to escape from antibodies. But in this case, escape from monoclonal antibodies. So going across the slide, you have monoclonals directed to NTD, RBD classes 1, 2, 3, 4. And then going down on the slide, you have the original virus and all the subsequent variants. And the red means the virus has escaped from the antibody. And you see the original strain is susceptible to all the antibodies. But alpha, beta, gamma, and delta escape from some of the antibodies. But once you hit Omicron, every class of antibody is impaired. And as the Omicron subvariants evolved, you have more and more red. That means the virus become even more resistant to different antibodies within those classes. This gives you a picture. These are based on actual data, but let me give you a graphic summary. So this original spike is labeled wild type. It's on the left, right? And in infected people or vaccinated people, you have all these antibodies that attack the RBD and the NTD, and a little bit on the bottom. But the virus does not make mutations toward the bottom of the spike because it doesn't feel the pressure. That means those antibodies are not very effective in getting down to the bottom of the spike. The virus doesn't bother to change. But it does change on the top a lot. And so by the time you get to the first Omicron BA1, most of the antibodies are not working anymore. So you are left with a few antibodies that are still active. And now when we get to XBB, which is the next jump, you only have a few antibodies that could actually get through and neutralize the virus. And importantly, that SD1 antibody is knocked out by JN1. So basically, we're left with this one particular strong arrow. And so this is one spot where we still have a chance using existing previous exposure to the antigen. This is the only spot. Obviously, if you have a baby who's recently infected with the new variant, that baby could make antibodies to all of these once again. But for most of the people in the room, this is what we're dealing with. So let me tell you about our last antibody engineering program. So these two colleagues, Li Hong and Chen, actually isolated an antibody which we called 1977. In red is good, okay? And you see this antibody neutralizes SARS-CoV-2, all variants except one called JD1.1, which is actually not very common. So we're pretty happy with this antibody, but some of the strains show, instead of dark red, show light pink or red. That means the potency is not so great. And then it neutralizes, interestingly, sarbecoviruses that are more genetically more distant. So we're pretty happy with this, but can we make it better? Can we engineer it to make it even more potent and also broader? So what my colleagues did was simply to to look at the structure. So we solved the cryo-EM structure of the antibody bound to the receptor binding domain. And it is a class 1 antibody as we expected. So we took the residues that are in contact and say, and those residues are listed on the top left, and on the heavy chain, on the light chain of the antibody, and say, we make an alanine substitution at every one of those positions. And you could see that's what we did. And when it's red, it knocks out the antibody binding. When it's green, it actually improves antibody binding. And you see that R71A mutation is all green. And then next we look at the R71 position and substitute every other amino acid. And it turns out that all of them except for the K substitution gave us even better binding. And then we looked at the structure to say, well, what's so special about that residue? And it turns out that residue holds the part of the amino acid so that it's not very flexible. The the so-called CDR loops of the antibodies. And what the substitution does is freeze up the antibody. And look now, with the 3 variants, with the A or L or V substitution at position 71, you could see the improvement just by the color. It's more potent and it's broader, and it hits all the viruses that we have tested except for SARS-1, the original SARS virus. And we could solve the structure and basically on this slide simply prove that what we have done is make the antibody more flexible so it could accommodate. And now you could see we could actually get the modified antibody to bind to JD1.1 RBD. So it now is more flexible and actually could bind. So in the remaining few minutes then, I'm just going to tell you a little bit about something that's fairly new. We're tired of chasing after the virus. And also, we expect more pandemics to come, particularly as we have increasing population, increasing travel, increasing contact with animals. And today, we already have a pandemic of H5N1 influenza in birds. And we know only a few mutations are needed to convert that into a human pathogen. But everybody's saying, what is the next pathogen? Well, we don't know, and therefore it's really difficult to prepare for that. And so the question is, could we come up with something that will protect very broadly against many different viruses? And that's what I will finish with. So this is the work of 2 long-term colleagues, Maria Tsuchi and Huang Yaoxin. And what we have done is come up with a chemical entity that stimulates the immune system and can protect, in animals, 3 different viral infections. And this chemical is something we call glycolipid. And the compound that we're working on doesn't have a name. It's labeled 7DWA5. You could see it has a galactose head and 2 lipid tails. And importantly, this molecule binds to CD1D on the surface of dendritic cells and then presents it to invariant T-cell receptor on NKT cells and turns on the release of cytokines and triggers a cascade of immune reactions. So now I'm going to show you a bunch of animal experiments. So we didn't actually think this would protect. But we gave it a try. And the first experiment we did was in mice, and it's shown on the left schematically. So we administered this chemical, glycolipid, intranasally into the nose of the mice 1 day prior to giving the mice the virus, also intranasally. And then for 3 days in a row, we weighed the mice to look for weight loss, and then we sacrifice them, harvest the lung tissue, and measure the amount of SARS-CoV-2. And then if you look to the right, we compare glycolipid to saline as placebo. And you could see with saline, the mice lost about 10% of body weight, whereas those treated with glycolipid did not lose much weight. And if you now look in the lung tissue for the amount of virus further on the right, you see there's lots of virus, 10 to the 6 in saline-treated mice, but the glycolipid mice, there's about 1,000-fold reduction in the virus. So this was a real surprise to us, a pleasant one though. So we went on to see if we could work out the route, the timing, the dose, and the repeated use of this agent. So on the route, you see on the left now, whether we give it intranasally or intravenously, we have protection against weight loss, and we have protection against virus replication. Then the timing, you see, if we give it 1 or 2 days before, -1, -2, you see very strong protection. If you give it day -3, you have partial protection. So it lasts somewhere around 48 to 72 hours. But if you give it after virus infection, it doesn't really work. So it's a prophylactic agent. Now, in terms of the dose, we could go as low as 0.1 microgram and still get protection. And in terms of repeated use, we could give it 5 times in a row over 10 days and still have the same protection. So there's not some sort of burnout effect. And this experiment simply shows that we could get the same protection whether we are using the original SARS-CoV-2 or Omicron BA.5 or Omicron BA.1 or the Delta variant. Although the Delta variant, the extent of protection is not as great. And I think this is the most important observation. Here, on the left side, we're looking at normal mice or mice with interferon gamma gene knocked out. So in the normal mice, we see the same protective effect as seen before. But when you knock out the gene for interferon gamma, you could see saline and the glycolipid gave the same result. There's no longer any protection. So gamma interferon is an important mediator of protection. You move to the right, it's the same experiment, except on the right side of the slide, the mice are CD1D knockout mice. So CD1D is the receptor for the glycolipid. So if there is no CD1D, there's no protection. So it's a broadly immunostimulatory agent mediated through a very specific pathway requiring both CD1D and gamma interferon. So we wanted to see if this protection could extend to other viruses. And you could see here on the left, it's the same protection experiment, but not against SARS-CoV-2, but against a family from the paramyxovirus called respiratory syncytial virus, or RSV. And you could see the same protection against RSV. And then moving to the middle is the protection against influenza virus. And on the right is an extreme lethal challenge model against influenza also. So we think these are type of strategies that should be pursued much more to come up with agents that could block many different virus infections. So basically, we're exploring the innate immune system, which is our first line of defense, to see if we could confer much broader protection against many different respiratory pathogens. And we think agents such as this could be prepared so that should there be another pandemic that is spread through the respiratory route, it could be used as a prophylactic agent. So I've spoken for a long time. I've given you a very broad perspective on our work involving 2 different pandemics, but I won't go through another summary. It's late in the day, and I just thank you for your participation, your attendance, and I thank MUST once again for the great honor. Thank you very much.
Thank you. Thank you for your magnificent speech. We will now proceed to the Q&A session. Dear students, if you have any questions, please raise up your hands, and our staff will pass to you the microphone.
So thank you so much, Professor, for the fruitful talk for us. And I have a question about it. Because the HIV virus actually migrate from the genital area to the PS patches in the jejunum and the small intestine, is there any possibility that, because I've read some research about the diversity and also the richness of the microbiota in the gut actually is different from normal people and a HIV patient. So is there any strategies or novel intervention that can we use the microbiota for prophylaxis or the treatment to aid the treatment for the cocktail therapy?
If there is one, that's very promising. We'll be working on it. But in terms of microbiota, it's not an area of expertise for us. And yes, there could be differences in the makeup of the microbiota in normal individuals as well as those immunodeficient individuals. But so far that hasn't really provided a major clue as to how we might tackle HIV. I wish I had a better answer for you.
Thank you. Are there any students who have any questions? The student.
So thank you for Professor for the speech. So my question is, so the problem of the HIV which cannot be prevented from the vaccination is about the rapid variation. So my question is there any of the possibility for the 3D printer targeting the GP 120 and GP 41 in order to lower the cost, including time cost or the money, and even to desire a more precise vaccinations for the major HIV variation?
Yeah, it's a tough problem. As I said very briefly, the field has been working on this since the discovery of HIV in 1983-84. Any vaccine strategy you could think of has been tried without success, trying to tackle basically GP 140 and 120 and GP 41, as you say, but the viral diversity is enormous, and which starting virus or viruses which you use is a great controversial area in the field. I would say there's still enormous amount of work going on to move toward an effective vaccine. but I personally don't see anything really promising coming about in the near future. But it means that we still need to make the commitment to work on it. And in fact, it is through working on HIV vaccine that developed so many of the platforms, including the mRNA platform that is now applied so widely for COVID-19. So even if we are not succeeding with the HIV vaccine, we'll have many other benefits that will be derived from that.
Thank you. And the last question, so please pass your microphone to that boy.
So
yeah.
I have a question.
Okay.
Oh, thank you, Professor David. I'm a big fan of you. Can you hear me? Thank you, Professor David. I'm a big fan of you. Like, the question of mine is a little bit naive. Is like, we all know that Omicron have been a very big contributor in end the pandemic. The question I want to ask the feasibility of making a human-made virus that is likely to have high infectious but low toxicity, or there's any other ways we can accumulate the speed of the Omicron being funded, so we can also end the pandemic much faster than it used to be. Do we have a chance or like feasibility to make that kind of human-made virus vaccine like this?
Yeah. Nowadays, there's still lots of concern about re-engineering any virus because of the possibility the virus may have gain of function, may become more transmissible, may become more virulent. So that sort of research is not often approved, first of all. Second is, if you make a virus for whatever reason to tackle the viral evolution, it's a bit of a guesswork. It may or may not be right. And nowadays, years ago, we all got viral vaccines that were made from the original virus but attenuated. And these days, that's just not in favor, because as you attenuate the virus to make a potential vaccine, we all recognize that reverse mutations could occur to restore the virulence of the attenuated viral vaccine. So the field is not moving in that direction. Doesn't mean it's right, but it means that's not in favor.
Thank you. So this is the last question. So thank you, Dr. Ho, for answering the questions. We also prepared flowers for Dr. Ho. May we now invite one of our students to present the flowers. So let's take a photo. Please look at the camera.